Hello, I'm Gemma, one of the senior biomedical scientists in microbiology here at Wolverhampton at the BCPS Hub. If you want to come through, we'll take you on a tour of the department. We'll start down at the reception where all of the samples come in, and then we'll work our way through the lab. So this is our specimen reception area. This is where all of the samples that are taken for microbiology come into first. So every single sample has to be booked onto the computer system. That goes for whether we're testing it or even if we're not. So if we're not testing it, we still need to let the doctors know, that person who's requested it, that they're not going to get a result. So all of the samples either come up through the hoist from downstairs, their specimen reception downstairs, or a person will bring it. So that's a lot more common now with COVID samples. All of those are brought directly to the lab so that they don't get lost en route. So this is our main specimen processing area for the pre-analytical side. So we have a lot of support staff working in this area, getting all of those samples cultured, going through the systems. So this is our wasp analyzer. So we can see the samples going at this end. Obviously they're barcoded, so it can be barcoded right the way through and tracked. It brings it along on a little conveyor belt, and then it'll vortex the sample, it'll uncap the sample, and it'll inoculate it onto a variety of agar plates. So each of the agar plates has all of the nutrients that the different bacteria need for that particular type of sample. It will inoculate the plate, so we've got a nice standardised culture that goes, whereas when we're doing it ourselves manually, obviously some people might treat slightly differently to others. It'll incinerate the loop, so it gets it nice and hot to kill any of the bacteria that might still be on there, so that we're not carrying bacteria from one sample to the next. Each plate has its own barcode, so that we know what that plate is and which patient and which sample it relates to. You can see once it's done the sample, it takes it back, and there's a conveyor belt along the back which will take it to the incubator. So we can see we've got the incubators along the back, and then inside each of these is a camera. It'll take the image, and then that image can be read digitally. So at this point we've got that nobody has been exposed to anything so far, so it keeps that operator nice and safe throughout the process. This is where those images are then read. They can look at the plate and see, is this something that would normally grow from that particular site, or is this something which needs some further work, and we need to identify and work out what's causing an infection in that patient. So this is our mass spec, so it uses mass spectrometry to work out a profile for the different proteins that are within that particular isolate. Once it's got that profile, it can match it against the database, and it can give us an identification within 20 minutes. From there, we need to work out which antibiotics it is that we can use. So you can see we've got a variety of different microscopes depending on the type of sample that we're looking at. And along the back we've got the manual incubators. So again, we've got that option to incubate things offline. If it's something that either hasn't gone through the wasp, or if those incubators are down, we can put them in there instead. So this is our serology section. Again, we split it into automated and manual, but realistically nowadays it is all automated. And now I'll be testing for things like varicella zoster to check whether a patient is immune and won't get chickenpox, especially during pregnancy. For C. diff, which is particularly important in the hospital setting, so that we know that we've not got toxin present in those patients. This is where we deal with all of the waste, so sometimes it can be a bit smelly in here. So we've got the autoclave, so all of our waste, it has to be made safe. It's our responsibility to the public and to the community to ensure it's safe before it leaves the department. So it will go through the autoclave, it gets heated to a really high temperature. So these are our main molecular rooms. So nowadays COVID kind of trumps everything, and it's taken over the department to some extent because we do the PCR testing. So one of the rooms in which that's done is this room here. So we've got a machine that will prep it, a machine to do the extraction, and then it goes next door for the amplification. So this is where the amplification happens. So it goes through a set program, ramping up the temperature and cooling it down, allowing that PCR process to take place. So this room is where the chlamydias and the gonorrhea are tested. And at the moment, due to the use of other rooms for COVID, this is also where the HIV and Hep C viral loads will take place as well. So this room is where we do all of our urgent testing for COVID. So we've got the two types of instrument. We've got our Kefid Infinity, which does its PCR process the same, but it all works within one cartridge, and within 45 minutes we can get a result off. And then we've also got the Sambas, so it's a PCR-based approach, but it works in a slightly different way. So this is the extension that's been built as part of the BCPS move. And then as a late addition due to the pandemic, we got the COVID lab. So this is where the bulk of the COVID testing is processed. Thank you for coming on a tour of microbiology. We hope to see you again soon.